Hi guys, so we are left with the end section of proteins. Today we will be discussing protein folding. Now, as you know, proteins are extremely thermolabile, pH sensitive substances. They cannot work at varied pHs. So they have specific pHs because of their zwitterionic forms, and they have specific temperatures, which is the optimal body temperature in which they work perfectly. But the nascent polypeptides, which are formed via translation, are extremely susceptible, in fact, more susceptible to these fluctuations that happen within the cytoplasm of a regular cell. Therefore, these polypeptide chains need to be folded so that they can develop further hydrogen bonding, covalent bonding, and electrostatic bonds between their components, which will give it a certain amount of higher stability to be able to withstand the cytoplasmic environment, right? So that is what is protein folding. So now the primary structure or the primary chain of a protein is the linear sequence that we have. It is the polypeptide sequence. So because it is formed by joining multiple amino acid chains, it will have an NH end and a COH end. Correct? So that's your polypeptide chain. The peptide bond between each amino acid would have a spacing of 0.35 nanometers. These polypeptide chains are non-functional. They need to be folded before they can be sent away to their particular locations for functioning. Otherwise, they will be degraded or denatured down to amino acid stature because of different pH and temperature conditions of the cells. Now, the secondary structure, the first step into folding, there are two kinds of secondary structure. One is an alpha helix, so it is a right-handed helical structure, right? In which the first amino acid, so let's say these are the amino acid, okay? So there are 3.6 amino acids per turn. So if I were to draw it, something like this, right? So in one turn, you would have 3.6 amino acids. So what I'm trying to say is that in this particular helix, if this is the first amino acid and this is the fourth amino acid, there is going to be a hydrogen bond between them. The oxygen of the COH group of the first with the NH group of the fourth is going to bond and form a hydrogen bond. This is going to give this structure a certain amount of stability. Correct? So keratin, myosin, these are all alpha helical structures. Coming to pleats, so when I talk about pleats, they're basically sheets formed of more than one polypeptide chain. Now, if both of them have their N and C groups at the same site, and there is hydrogen bond forming in between, then they become parallel pleats. And if they have their NH groups juxtaposed with the CO groups, then they become anti-parallel pleats. Yeah, okay. Both of these structures will obviously add stability to the polypeptide chain, and there can be more than one polypeptide chain associated with one secondary structure. Correct? Now, tertiary structure refers to a 3D shape. So, when multiple foldings where beta sheets and helices have come together to give it a 3D modification, we call it a tertiary structure. We have to remember that proteins who have attained a tertiary structure are functional. So, secondary proteins are non functional. Proteins start becoming functional from their tertiary structure. More than two tertiary structures coming together, for most proteins, the most functional structure then becomes the quaternary structure, which is the end stage of protein folding. For example, hemoglobin is a quaternary protein, right? Now, depending on how many and what kinds of polypeptide chains are present, proteins can be further classified. So, proteins can be classified into, I'll erase this. Proteins can be classified into homomers and heteromers. Okay, polymeric or monomeric. Yeah. So homomers, same kind of polypeptide chains. Heteromers, different kinds of polypeptide units. Monomeric, one polypeptide unit or chain making one protein. Polymeric, more than two or more. So 2 to 10 we can also call it oligomeric, and up after 10 polypeptide chains we can call it multimeric or polymeric. So for example, uh, myoglobin is monomeric, but insulin is dimeric. It has two different kinds of chains, right? So hemoglobin, for example, they are all polymeric chains. I hope this much is clear. Other than that, we have another class of protein known as the first class protein. So proteins which are made up of all essential amino acids are called first class proteins. I've given you every detail that you might require for NEET solving. In next few lectures, we are going to solve a few questions as well. I hope this was clear. Thank you.